We all know that most of our ELISA kits are about protein biomarkers, protein or peptide biomarkers. And we all know that it is impossible to develop and make high-quality ELISA kits without having high-quality raw materials. So tomorrow in the tour we will show you that we have also a recombinant protein department, and but I think that the most key components in immunoassays or in ELISA kits for protein biomarkers are highly specific and affinity antibodies, and Helena will tell us something about our best sold antibodies and antibodies which we use for the production of IVD registered kits.
Thank you for introduction, Vladimir. As Vladimir said, I will give you really brief information about some of our products, and then I will focus on one of them. So, all of the antibodies I present here are originally produced by BioVendor. In this table, you can see antibodies which are involved and also verified in our IVD ELISAs. These antibodies can be offered as a match pairs, most of them, and also they can be included in different IVD applications. We offer more antibodies which are included in our ELISAs, but they are not listed in here, and if you want to know more about them, you can tell me. I will be happy to give you this information. Here are listed our best sold antibodies, and here is information about areas in which these antibodies can be applied. You have these informations in your materials, so you can study them. Our best or our most popular antibody among our customers is rabbit polyclonal anti-periostin antibody. In this presentation, I would like to point out the big potential this antibody has. And I will start by focusing on the protein against which this antibody is targeted. This protein was first isolated from a mouse osteoblast cell line, and it was called osteoblast-specific factor 2. Later, it was found that it is primarily expressed in periosteum, and thus it was renamed periostin. Under physiological conditions, it is involved in processes like development, growth, remodeling of different tissues, and in normal adult tissue, it is expressed in low levels and overexpressed under pathological conditions. As you noticed in the title, periostin is described as multifaceted protein. It is because it plays many different roles in human biology, and these roles are given by its ability to act in two ways. At first, as an extracellular matrix protein, it can bind other components of extracellular matrix, and it's like collagen 1, fibronectin, and others. And this way it affects maintaining of tissue structure or collagen cross-linking or generating fibrosis. At second, it can act as matricellular protein. This is a group of proteins, of extracellular matrix proteins, which are able to bind to their receptor on cell surface. This way, periostin can affect modulation of cell adhesion, migration, proliferation, and differentiation. Today, I will mention only some of the roles of periostin, not all of them. The first on the list are renal diseases, and one of them is infamous chronic kidney disease. My colleague will talk about these diseases later in connection with ELISAs. I will just remind that these diseases are characterized by gradual loss of kidney function, which can finally result in dialysis or kidney transplantation. And there is need for new biomarkers which can early diagnose the kidney diseases or which can serve as a targets for therapy. And in this regard, periostin seems to be very promising. It is expressed in low levels in healthy kidneys. However, after renal damage, it is highly overexpressed. And its expression is localized in injured cells, which means it's specific. And what is important for good biomarker, it is secreted and it can be detected in body fluids. Also, the levels of periostin are well correlated with CKD progression. All of this makes periostin to be a good diagnostic biomarker. It was to mention that in animals which were treated with inhibitors of periostin, they were protected from development of severe form of renal diseases, which indicate that periostin can be used also as a target for therapy. If we proceed to the end stage of this disease, the patients which has a transplanted kidney has in fact better quality of life and longer survival than patients who are dialyzed. But there is important to have good markers which can reliably predict graft survival. Very recently it was published the study on patients with kidney graft biopsy, with kidney transplantation, and here was shown that expression of periostin was higher in patients which later experienced graft failure. And there was significant relationship between periostin and graft loss. And also statistic analysis revealed good predictive value for identifying patients with graft failure. So periostin can be investigated as a predicting biomarker. On the immunohistochemical staining, which is taken from this study, is shown staining of periostin on kidney graft biopsy, and it was made with our antibody. Periostin is also involved in inflammation and allergy. Here belongs asthma, which, as you know, is a common long-term inflammatory disease. Usually patients with asthma are treated with inhaled corticosteroids. However, about 5 to 10% of patients are insensitive for this treatment and requires another therapy. In this moment, serum levels of serum periostin can help to distinguish what type of therapy will be suitable for the patient. If the levels of periostin are high, it is possible to use antagonists of type 2 immunity response. In case of low levels, it indicates that there is necessary another therapy. So periostin is investigated as a marker which can predict efficiency of specific asthma treatment. In this graph, you can see the group of patients with high levels of periostin, which has a better response to the antagonist of type 2 immunity than group C, which is low periostin levels, group of patients with low periostin levels. So another area is cancer. Periostin is involved in development, progression, and metastasis of cancer. And in the study on prostate cancer patients was shown that periostin was overexpressed in more than 80% of cancer patients. And also that the overexpression was limited to the tumor tissue. And what worth noting is that high expression of periostin was significantly associated with shorter survival of patients. On the immunohistochemical staining, you can see periostin expression in different grades of prostate tumors. The similar findings as in a prostate study were made also in many other carcinoma cancer types. And general conclusion from these studies is that the high periostin levels are usually associated with a more aggressive tumor behavior with advanced stage or poor prognosis, which makes periostin very promising prognostic biomarker for cancer diseases. I would like also mention that periostin is involved in resistance to some types of chemotherapy, and it can be used as a target to overcome this type of chemoresistance. And in some animal models was shown that when the animals were treated with antiperiostin antagonists, they show lower growth of primary tumors and they had a lower number of metastases. So let me summarize the advantages of periostin, which makes him a promising biomarker for different diseases. It is possible to measure it in a plasma, urine, and other body fluids. There is a significant difference between expression in healthy tissues and under pathological conditions. Expression levels correlate with the stage of the pathology or with the prognosis. And targeting of periostin can positively affect treatment of disease. As you can see, periostin has a big potential for use in many different research areas. And I think that we can expect also growing interest in the antiperiostin antibodies in general. So, why offer our antibody to your customer? This is a high-quality antibody. It is specific, it's user-friendly, it's reliable, and its usage is confirmed by many of our customers. Interestingly, on website of the biggest OEM distributors, this antibody is the best rated among antiperiostin antibodies, and on another distributor website, it is stated as a best sold antibody among antiperiostin antibodies. There is more than 100 scientific articles which describes using of our antibody. In these papers, the antibody is referred to as BioVendors or OEM product, which is reason why you cannot find all of these references on our website. Most of the scientific studies concerns oncology, and then it is followed by cardiology, renal diseases, and other. I didn't list it all of the areas in here. And when we look at the method which is in which our antibodies are used, the most frequent is immunohistochemistry followed by Western blot. So as you could see, our antibody can be used in various research areas, in various clinical studies, and I really believe that it can be interesting also for your current or potential customers. So, thank you for your attention, and if you have questions.
So do you have any questions to our antibody productions?
Do you have Periostin ELISA?
Sorry?
Periostin ELISA?
Yes, my colleague will talk about.
How do you validate the lot-to-lot continuity? Do you test your antibody each production in?
Yes. No, in ELISA we test electrophoresis titer or Western blot.
Strong computer. Which company is strong competitor with antibodies?
Competitor. Do you mean OEM distributor? No, competitor. I know that R&D sells, but they offer monoclonal antibodies, and this is polyclonal antibody, so it's different. And in fact, I don't know.
Which kind of customers are buying it most?
Well,
Mostly it is research customers, research institutions. Yeah.
Translational research? Diagnostic research?
I think they're both of them. Yeah. I don't have the numbers in my mind, but something like that. Yeah.
It may say that periostin is non-specific marker because it's a patient covering some oncology diseases, has problem with kidney, with some cardiovascular. It depends on level periostin. It's difficult to say which problem.
I think that usually the specificity is it is specifically overexpressed in the tissue, and
Tissue,
yeah,
exactly.
so it is good. For ELISA, it's problem a little bit. It can be problem because there can be more influences, more effects which can affect the levels. For example, for the asthma, in a very recent study, they shown that it can be detected in airwaves condensate, and that there it is it has better correlation or yeah.
I would say that it is not used for diagnosis or differential diagnosis. It is more used for the monitoring of or for the staging of the disease, already identified disease, I mean, or diagnosed disease, and for the prediction of therapy, etc. Yeah, so it is not the marker which you use for make the diagnosis. But if you monitor somebody with already diagnosed cancer, then the changes, the dynamics of the change or kinetics of the change of periostin can tell you something about disease progression or regression.
Like additional marker,
Yeah.
For
Yeah.
tissue,
Yeah.
especially for prostate.
Yes.
You can use it in combination with the
Yes.
specific markers for the monitoring of the therapy, for the monitoring of the progression and so on.
What about your selling products best for antibodies or just ELISA periostin?
Antibody. We
Antibodies.
are much more successful in this case with antibodies than with
Yes.
antibodies.